All right. So today I will talk to you about some of the research that we have been doing in my lab for a long time about cellular senescence, and recently in the consortium that Lisa mentioned. But a little bit of motivation on why do we study aging. This is an important problem because, as you can see in terms of projections, aging is a problem that is going to just become a fundamental thing that we need to address in the future, because the population is at high risk of diseases that are associated with degrees. As you can see here, the projection is that by 2034, we will see a crossover. There will be more people that are 65 or older than young people. And this is the projection for the stratification with age, 2060. And so, as you can see, this part, starting at 50 or 60 and up, this is where all the chronic diseases have become really prevalent. So this will become a burden for our health system, and we need to find a solution for that. And we can try to cure every single disease, which is what we're already doing, but we already know that aging is a key risk factor for disease. It's probably the single most important risk factor for a wide range of chronic illnesses, including diabetes, heart disease, neurodegenerative diseases, cancer. So in the field of the biology of aging, our mantra is that if we find intervention that can slow aging, those interventions have the potential to slow down the onset of these chronic diseases, prevent them, and not just one of these diseases, but many diseases. In the context of the biology of aging, over the years, we have identified many hallmarks. As you can see here, this is a slide that if you hear any aging talk, you will see over and over. And these are the main hallmarks. In 2013, this was published. There were fewer hallmarks. We found more of them. But today, I will focus on two of these hallmarks, which are a big part of our research program in the lab. And this is cellular senescence and chronic inflammation. What is cellular senescence for the people that are not familiar with it? Cells go through the cell cycle, and they usually end up divided into two new cells. But if they accumulate damage, there is a control system that triggers the permanent exit from the cell cycle. And this is thought to be evolved as a protection in terms of preventing damaged cells from dividing. And we will see that these cells can accumulate genetic mutations, and they could lead to cancer. So this is a protective mechanism. So that is the rationale that people have come up with in terms of why this evolved. If we look at what these senescent cells do in terms of what they change into, they are characterized by several hallmarks. And the hallmarks have been discovered over the course of many years of study. One of the first ones is cell cycle arrest, as you can see here. Of course, I told you they're permanently arrested, so they upregulate some of the controllers of the cell cycle, typically P16, P21, and they downregulate genes that are involved in the cell cycle progression. You have also changes in the nucleus, and I told you most of senescence is triggered by damage. So you can see the appearance of DNA damage foci in the nucleus of the senescent cells, and this triggers the DNA damage response and senescence in general, as you can see here. The DNA, the nuclear changes that you see are also the level of the epigenome. And what we can see is a loss of heterochromatin, which is the chromatin that keeps a large part of the genome silenced. And these silenced regions harbor elements that we don't want expressed, transposable elements, for example, that can lead to genomic instability. You tend to lose these epigenetic marker, heterochromatin, at the size triggered typically by DNA damage, and these elements start to be expressed. And as you can see here, this instability in the nucleus, triggered by DNA damage and fragmentation of the nucleus, plus the activation of these elements, leads to the appearance in the cytoplasm of DNA species. These are called CCF, chromatin fragments that are in the cytoplasm. So cytoplasmic chromatin fragment, as well as these line elements end up in the cytoplasm as well. This triggers a protective mechanism that we have from viral infections. So if we detect DNA in the cytoplasm, C gas in this pathway activate the interferon response. These endogenous species that are found in the cytoplasm as a consequence of DNA damage in the cell trigger this interferon response. So these cells become a source of inflammation. Other hallmarks of senescence are the upregulator of anti-apoptotic pathways. Although these cells are damaged, they still survive. They're fully metabolically active, and they stay in place, and they can survive in your body for a very long time. And so there's this upregulation. Importantly, that's one of the key elements of these cells. You have the secretion of inflammatory factors. Interleukins and other chemokines are secreted by interferon triggered by these CCF, for example. And that's a big part of the story that we'll talk about today. And then there are other hallmarks like increased lysosomal contact and beta-gal activation. This is one of the markers that we use in order to detect senescent cells in cell culture and in tissue. And you also have metabolic adaptation, and mitochondria become dysfunctional. They start to produce more reactive oxygen species. They also release their mitochondrial DNA in the cytoplasm, contributing to the interferon response. And they have some cell surface markers that are enriched in these cells and that are used by a lot of groups in order to target them and identify them. So this is kind of an overview. But what is their role in aging? Their role in aging is that under normal circumstances, if you have a functional and active immune system, these SASP, senescence-associated secretory phenotype, which are inflammatory signals, alerts the immune system, which comes and clears the senescence. So it's the typical model is damage, inflammation, which is a signaling for the immune system to come and clear the senescent cells. So there is a balance between appearance of these cells and the immune system ability to clear them. What happens when you're young is this balance is maintained so that you have a very low level of senescent cells in the body, typically from 1 to 5%. As you age, the number of senescent cells becomes more and more prevalent in the tissues because the immune system has less ability as it declines to clear them. And that creates a chronic inflammation in the tissue, which leads to functional decline of the tissue. They're not only involved in aging, but they're also involved in specific diseases. And this is like an overview of all the diseases. As you can see, they're across many organs and many types of diseases. Fibrosis is one of the main diseases that are triggered by senescent cells. But of course, there's cancer. Some people are looking into reactivation of senescent cells, which are already damaged at the level of their genome, and it could actually be a source of cancer. They are involved in metabolic disorders with senescent cells in adipocytes, fibrosis in the kidney, in the heart, in the liver, osteoarthritis, and also neurodegenerative disease. There's a lot of activity in studying senescent cells in neurodegenerative disease because some of the senescent cells appear to be glial cells that can then propagate their signal to the neuron and create an inflammatory response, which is associated with Alzheimer's disease, Parkinson's disease, and others. For the longest time, senescent cells were considered, before they were found so extensively in tissues, an artifact of cell culture. The way they were discovered was by Hayflick through passaging fibroblasts for a long time. And he did this over and over. And after a certain amount of passages, cells stopped dividing. They didn't die, they stayed there and divided. Of course, this is what is called replicative senescence, which is due to the shortening of the telomeres, which is then recognized as a form of DNA damage, and it triggers a form of senescence called replicative senescence. Over the course of the years, the SASP was found to be a critical component of senescence, and they were found in many tissues. And the idea was if they're really associated with disease outcomes and aging, they could be a therapeutic target. And the breakthrough came with this paper in 2016. And what they did here, they used one of the markers of senescence, which is P16, and they engineered a mouse which could induce apoptosis in specifically cells that have an active P16. When P16 is active, these cells are induced into apoptosis by the use of this compound AP.187. This was a breakthrough because, as you can see here, these are 22-month-old mice. The one on the right is the one that was treated with this protocol. And the one on the left, same age, was not treated. So you can see that even from the point of view of their appearance, it looks much younger. It doesn't have the typical aging signatures of the one that was the control mouse. And in looking at their survival, there was an increase of 27% in the survival for the treatment. So this actually puts senescence clearly on the map as a therapeutic target for aging and many of the diseases, because as you can see, the phenotype was much better for the mouse that was treated. This has become a big industry right now. That was a genetic intervention to clear senescent cells, but over the years, people have developed many approaches to clear senescent cells. These compounds are called senolytics, and they target different part of the pathways that are involved in senescence. The first ones that were developed, generation 1, work on what I said before, this ability of the cells to survive apoptosis. So inhibitors of the anti-apoptotic pathways. And so these were compounds that derive, these are chemotherapies that were already demonstrated to work with cancer, and they were also used as senolytic, and they work pretty well, although they have side effects. And so the whole game now is to try to find senolytics that are as specific as possible to senescence and have very little side effects. On the other side, what I told you was the big problem of senescent cell is not their presence, it is because they are inflammatory. So this is the other part of the approach to chemotherapeutic with compounds that are called somomorphics and target the pathway that triggers the SAS, the senescence-associated secretory phenotype. And right now, the jury is still out which one of these two approaches is the best one. And many people say that probably a combination of the two is the way to go, because in some circumstances, xenomorphics are less toxic, are easier to develop, but senolytics ultimately will get rid of the problem. What I will talk to you about today, as I said, will focus on let's start with the concept of the SASP and the inflammation. So the SASP is this signal that is released by senescent cells, and typically should be a signal that is targeted to the immune system for clearance. That's not all it does. In 2013, the lab of Masashi Narita, Jesus Gill, developed a protocol to see what happens when healthy cells, these are fibroblasts, in a co-culture system are exposed to the SASP that derives from senescent cells. So as you can see here, you have senescent cells, and these are non-senescent cells, healthy cells. So there is a way in cell culture in which you can induce senescence that is called oncogene-induced senescence. And what you do is you take a gene like RAS, for example, and you overexpress it. That leads to overproliferation of the cells. the replication force collapse, and that leads to a lot of damage and self-become senescence. So you could induce selectively senescence only in the subpopulation. These were induced into senescence, and these were the control cells. So what they demonstrated is that the cells can propagate the senescent state to other cells. So a side effect of the cells, which is supposed to just alert the immune system, is also that it can propagate the state. This is called paracrine senescence via the cells. And it turns out that this is not the only way in which you can propagate the senescent state. So this is work that we started a while ago. This is a collaboration that we did, and we stumbled upon this actually by chance. I'll tell you what happened. So this was a collaboration with Tamir Chandra. At that time, he was at the University of Edinburgh. Now it's at the Mayo Clinic. And we were trying to characterize the heterogeneity of the senescent state in cell culture. Early days of single-cell transcriptomics. So what you will see here is very low number of cells. At that time, we couldn't assay thousands of cells. It was tens to hundreds of cells. So the cells were induced into senescence with the system that I told you about, this ER-RAS system in which you get into non-contained senescence. And we looked at the time course of the cells, and we wanted to see, do they create a uniform transcriptional uniform state, or do you see subpopulation of cells in slightly different states? What we were surprised about is that we saw two populations over the course of time forming. And you have this one that has a signature that is typical of senescent cell, OIS stands for oncogene-induced senescence, these red ones here. But there was also another, which is a secondary senescence. And you see here that there are circles and X's. So what happens is when we look at the ERS gene engineering those cells, we have the ability, because we sequence it, to see if the ERS gene was really active in those cells or not. So we stratified them with those that has a system that could induce them into senescence, and those in which, for some reason, that system did not get activated. The X is the one that did not get activated. So these cells, X, could not be primary senescent cells. They have to be cells that become senescent because of the influence from the others. And we said, okay, this is paracrine senescence. They were not primary senescent cells. They're probably secondary senescent cells. So the paracrine senescence that it was already demonstrated as a means. The problem is when we started looking at the signature of their transcriptional profile, they didn't look very much like the paracrine senescence that it was described before. We knew at the time that there was another way that people have demonstrated that we can induce senescence, and this is through the notch pathway. So if you upregulate notch, this triggers the inhibition of the standard RAS-induced senescence pathway and an activation of the TGF-beta notch-induced senescence. When we compared our transcriptional profile for these cells to this data set that was already published, we saw that we had a very high significant overlap between the two. So this told us that with high probability, these secondary senescent cells did not form primarily through the paracrine pathway, but they formed through the notch pathway. And then we went down the path of trying to see this in liver, and we're still working on that. But the indication is that there is a contact-based way of inducing senescence, which had not been identified before. And so we have two ways of inducing senescence right now, one through the paracrine and one through contact, which we call juxtacrine-induced senescence. This was as I said, this was a serendipitous finding with a data set that we didn't expect it would behave this way. So we continued the study and said, okay, is this something that can go beyond oncogene-induced senescence, and it can be seen also in other forms of senescence? So we did a study in which we tried to look at different forms of senescence. We redid the oncogene-induced senescence. But this time we clonally expanded all the cells. So we knew that every single cell has a natural RAS construct. So that excluded the fact that some of them could have it and some could not. We used a replicative senescence, which is what I described at the beginning. We pass a cell many, many times. You get DNA damage at the level of telomeres, and they become replicative senescence. We also used another very common way of inducing senescence cell culture, which is through the administration of a topocyte. Topocyte is a topoisomerase 2 inhibitor. It inhibits the ligation domain of topoisomerase 2. So during the cell division, topoisomerase can chop the DNA to resolve the knots, and it cannot ligate it anymore. So that creates a lot of DNA damage. And of course, we use the proliferating as control. We use the more modern 10x single-cell transcriptomics, which allowed us to assay thousands of cells. And when we looked at the results, we saw that indeed these are the proliferating cells, and these are the oncogene-induced senescent cells. We could see two populations of these cells, even in this very genetically uniform cell culture. And when we look at the cells in cluster 2, which is this cluster here, with respect to cluster 1, we saw upregulation of the main markers that were also upregulated in our paper from 2019, which indicates that not-induced senescence is involved in these cells that become secondary senescent cells. The same happened with all the other two forms of senescence. So DNA damage-induced senescence is the etoposide, and also the replicative senescence, two clusters. And if we look at all of these markers, which are the same, are involved through the activation of TGF-beta and relate mostly to collagens and other markers that are part of the activation pathway for TGF-beta. This was an indication that all forms of senescence that we studied, we haven't done other forms of DNA harvest, for example, through gamma radiation, but this is something that we're going to do soon as well. They all form these two clusters of primary and secondary senescent cells. Why is this important? Well, first of all, it indicates that senescent cells are not uniform in their phenotype. There's a primary and there's a secondary. Does that matter? Well, if we treat these cells, at least in cell culture, with one of the senolytics that was the main senolytics at that time, navitoclax, which is one of the inhibitor of BCL2, so it was used very much at the beginning of the senolytic wave of compounds that were used. Here, what we did is we essentially treated with navitoclax the etoposide-induced senescence compared to the normal cells. And we did, again, single-cell transcriptomics. So we looked at the leftover cells in this dish here that were not affected by navitoclax. And of course, here this is just a control. So we had two controls and then a control for navitoclax on cells that were not induced into the senescence. What we saw was that, we did that both with etoposide and with oncogene-induced senescence, so two forms of senescence. We just didn't do it with replicative senescence for the obvious reason that takes about 6 months to do that experiment, because it takes about 6 months to get into a replicative senescence state. When we looked at the leftover cells after navitoclax induction, we saw that the vast majority of the leftover cells are the primary senescence cells. So it looks like navitoclax tend to work more on those cells that have a notch-like induced senescence profile with respect to the primary senescence profile. This could be problematic because if you look at their SASP, their secretory phenotype, the primary is highly inflammatory. The secondary is less inflammatory. So it looks like navitoclax is working very well on things that are probably not the primary target that you want to remove. This was, an indication that when you develop these senolytic strategies, you also need to understand what type of cells are you actually removing. And within the SENET consortium, it is becoming really clear that the senescent state is not unique. There's many states. There is this concept of xenotype, which is affected by the cell type, for how long the cell has been in senescence, because we know that over time the state of the cell can change, and the trigger, what triggers senescent cell. So those covariates need to be taken into account when developing treatments to that try to get rid of senescent cells, senolytics, or try to remove the SAS through xenomorphics. So, in conclusion, for this part of my talk, we talked about different forms of propagation of senescence. You have the initial one that was discovered, the paracrine senescence with the SAS. And then the justacrine senescence, which is the one that I described in my talk, in which you have a notch ligand interaction that triggers the activation of notch and the activation of TGF beta collagens and those other growth factors in our bacteria. But in reality, in the cell culture, what you have is probably composite secondary phenotype in which cells are exposed to different degrees to SAS and to the juxtaposition. So it's not one or the other, it's probably a complex combination of the two. And in order to sort this out, we started, ultimately we want to see if this model happens in tissues, but we continued our studies in cell culture. And in order to understand the contribution of these two components, at least in cell culture, we performed a time course. So this is very similar to what you've seen before, a topoisomerase-induced senescence DNA damage. But this time we profiled what happens over the course of time. As you can see here, we profile them from proliferating. We also use quiescent, and I'll tell you in the second part, we use quiescent. Day 1, day 3, day 7, and day 14. By day 14, they are pretty much almost all senescence. We use different stains to identify that, such as the beta-gal, S8 beta-galactosidase stain, which indicates the lysosomal activation, and also we are using incorporation to demonstrate that they don't divide. So by this time, we pretty much are almost all senescent. And this is the UMAP plot. Each one is a cell, and as you can see, they progress from proliferating all the way to the day 14. In order to answer the question of the contribution of primary versus secondary, we used the previous data sets that we had in which we could discriminate between primary and secondary. And we developed a machine learning classification model that was trained in those data sets and could achieve 93% accuracy in those data sets. And so it can discriminate between non-senescent, primary senescent, and secondary senescent. Because these three states rely on primarily the difference between dividing cell and non-dividing cell, we also included the quiescent cells as a control because we didn't want to call something senescent if it was just quiescent. So this one was a control. I'm pointing this out because this will become important in the last part of my talk. But for now, it's just a control that we use to develop this classifier. So now we have something that if we feed single-cell transcriptomics, each individual cell, what is the transcriptional profile, we can find with high accuracy if it is a non-senescent cell, a primary senescent cell, and a secondary senescent cell. And this is what we found. So over the course of time, at the beginning, they are all non-senescence. There is a little bit of senescence in every culture, cells of damage, even in the normal culture. And then you see that the reduction of the proliferative cells in the non-senescence and a progressive increase of the senescent cells. But what was interesting is that the secondary one spiked all the way to day 7. And then between day 7 and day 14, the things completely flipped. the primary became predominant, and then you have the basal level of the secondary. So there is an initial wave of secondary-like state and a steady increase of primary senescence. Now that we have this distribution, we can ask what happens during this spike in secondary senescence between paracrine senescence and justacrine senescence. And to do that, we developed another class, but this time we didn't have single-cell transcriptomics data for the paracrine because we haven't done the experiment that we should have done it. But we could use the main markers that are the genes that can best discriminate at a level of bulk between the justacrine senescent cells and the paracrine senescent cells. So we identify what are the genes that are the best discriminated between the two forms of secondary senescence. And we define a score for each cell to be more paracrine-like or more justacrine-like. And this is what happens in terms of the paracrine versus justacrine score over time, from day 1 to day 14. You have an early wave of paracrine senescence, followed by a steady increase in justacrine secondary senescence. So the model that we have right now is you have a lot of cells become primary senescent cells because they're induced into that. They start to secrete a lot of SAS factor. And the cells that have not made it into a primary senescent cell experience that, and they become senescence themselves, but mostly through a paracrine path. But ultimately, when the thing stabilizes in cell culture, cells are in contact with each other. And that's when the justacrine senescence establishes these secondary senescence that surrounds the primary senescence. We haven't done the imaging studies in cell culture to demonstrate that we have a model similar to this yet, but we decided instead of doing it in cell culture to try to see if this model happens in tissue. And so what I will describe to you right now is a collaboration within the CENNET consortium with Jian Ma, who's a computational biologist at CMU, and Stephen Wang at Yale, and he's the co-developer of one of the spatial transcriptomic techniques called MERFISH. And Tianxi Yang is the postdoc in Stephen Wang's lab that we call the experimental work. And so here we decided to use young and old mice, 3 months old and 18 months old. And I realized that if I talk to people that work with aging in mice, 18 is not really old. Let's put it like a older adult. And then we took a liver and we devised the panel. So MERFISH is a technique in which you can assay for each cell a panel of genes. And this was a panel that we designed together that contained 508 mRNA species that we can image. So for each cell, you will have the measurement for each one of these species. We used a panel that contained 167 mRNAs to define liver cell type, but also through literature curation and through the curation of the data sets that we have, we defined a senescent library with all the markers of senescence we thought were important for the discrimination between primary and secondary senescence, plus others that are the standard senescent markers. And what we did, I will not go into the detail of this. If you talk to anybody in the senescent field, they tell you that it is really difficult to define a senescent state, even if you have many, many markers. And so here we use the technique in which we use the panel of senescent marker that I described here. And we looked at those that were changing with the significantly with age between the two ages, 3 months old and 18 months old. Those were those that are most informative. And for each cell type, because based on the other genes, we could assign each cell to a specific cell type. We devised a scoring system for them to be senescence, and we call this the SEN score. So as you can see here, the color code for each cell in this part of the liver is reflecting of the degree of activation of markers of senescence in that cell type. And then we knew the threshold, and we said anything that is above a certain threshold, we call it senescent-like. We don't call it senescence because we don't know if it is senescence. We cannot take that cell and put it in culture and see if it divides or not. So it's senescent-like or has markers of senescence. And that's where we can start to see patterns of senescence-like cells in the liver. And the senescence score showed that overall in each cell type that you can see here, fibroblasts, hepatocytes, HSCs, and so on, was increased independently of the cell type. This is expected because many of these genes are actually inflammatory genes. So it's picking up not only senescence if you don't threshold, but it's picking up also a lot of cells that have some inflammatory. We also confirmed that there are morphological changes in these cells that are set as senescent-like. We developed an AI that can detect senescence based on the remote morphology. And as you can see here, we extracted a lot of morphological features like homogeneity of the signal. This is based on the DAPI signal in the cells that were selected as senescent in the merfish. And some of the features that were extracted are actually things that we have observed also in cell culture. For example, the fact that I mentioned has to do with how regular the periphery of the nucleus is, and it becomes less regular. In senescence, the area, the nucleus become bigger. So this recapitulated some of the things that we have seen in cell culture. And then we looked at the cell type specific senescence. So we identified senescent cells in the aging mouse liver, and we said, what are the cell types that show mostly senescent cells? And as you can see here, in terms of composition, the vast majority was hepatocytes, but also those are the most abundant cells in the liver. And what they showed is, again, an increase in nuclear size, a significant increase, right, is significant here, across all the cell types. A niche type diversity score that was decreasing, meaning the niche in which you see these cells tend to have less diversity in terms of cell type. So there is a level of change that is local to where these senescent cells are. And also you see a cell identity score that was decreased, meaning the cells that are in proximity to senescent cells tend to lose their cell identity. And they start to have expression of genes that we would not expect in that cell type. We also looked at their pattern in terms of distribution of these cells across different regions of the liver. And we saw that they appeared in two different forms. Some of these senescent cells were randomly distributed across the tissue. Others were in a situation in which, when we look at the senescence score, it's strong in the middle and gradually decreases as you move away from it. And we did a quantification, and we saw that jump happens at about 10 to 20 micrometers from the core of where the senescent cells are. When we look at signals like the notch signaling, that's where we were going with this. It's like, are these the cluster that we expect from what we demonstrated in cell culture might happen? We had mixed results. Essentially, we saw that yes, we can see some of these senescent cells in the center and intermediate non-senescence around it, or borderline senescence, and the distals have a very low senescence score. But when we studied these cells that are next to the primary senescent cell, not all the cell types showed notch activation. It looks like the cell type specific notch activation is typical of neutrophils, HSCs, and fibroblasts, but not others. So, for example, hepatocytes don't show this pattern that indicates potential justification of the primary senescence. So it's possible that the susceptibility of different cell types to paracrine versus justacrine is cell type dependent. So the conclusion from this is that there are dual modes of transmission. Senescence can propagate through paracrine or justacrine senescence. There's a temporal sequence in the dynamics that suggests that the initial paracrine wave is followed by the justacrine stabilization. But as we can see in tissues, that may be only certain cell types, not all of them. There's a tissue level organization in which tissues form these clusters of senescent cells. And I have to say, I was at the Senate meeting this week. Many other groups are now using similar methodologies to look at the spatial distribution and AC clusters. So this clustering of senescent cells is becoming a hallmark of aging. Cell type dependencies, as I said, the justacrine seems to be just sort of cell change. In the last part of my talk, I want to give you some preview of some results that are very recently developed in my lab. And I never thought I would start working on quiescent. I thought that the only form of cell cycle that I was interested in was senescence. So as I said, when you exit the cell cycle, if you don't re-enter the cell cycle because you're damaged, we call it senescence. But you can induce quiescence in different forms. And this is taking you to this G0 state in which you're metabolically active, and you can re-enter the cell cycle under the right conditions. And typically, there's two ways of doing this in cell culture. One is serum starvation, and then you put them back with the serum in the cell culture. They start dividing. It will take a little bit of time, but they're happy. And then you have contact inhibition. So if you don't split your dishes, the cells stop growing. So those are the two methodologies. In the quiescent group that we use for that development of the machine learning, and we use this as a control, we wanted to be extremely sure about them being quiescent, so we used both. We took them to contact inhibition and then we removed serum. So they were certainly quiescent. One thing I didn't tell you is you can use the machine learning to discriminate between the different states, but you can also look inside the machine learning black box and say, what are the genes that are used to discriminate between senescence, non-senescence, primary versus secondary? And there's a technique that's called SHAP. I'm not going to go into the details because it doesn't really matter here for the message I want to give you, but we looked at, these are, for example, the top genes that can discriminate between senescence and non-senescence cells. We were extremely surprised not to see a lot of the SASP here. We would have expected that SASP was like one of the major signals that can discriminate, and we didn't see it. And we tried to wrap our heads around this for a while until we started actually looking at the data, which is something you should always do. We plotted the violin plots for some of these SASPs, or IL-6, IL-1 beta, and others. And look at what happened here. IL-1 beta was through the roof in terms of expression in the cells in these quiescent. And these are two replicas of the quiescent in these quiescent cells. And so it was IL-1 beta. And we said, well, maybe this is an artifact. These cells are actually senescence. We just induced quiescent, but we were so harsh that they became. So we removed anything that had P16 or P21, that are the two major markers of senescence. And we looked at all the CDK inhibitors. They were, like, if we look at the quiescent, they didn't really have much. There was a little bit of P21, so that's why we removed all the P21 positive. And still, even after removing those cells that could have been potentially senescent, we see this high activation of IL-6 and IL-1 beta. So, well, this is just one data set. It is the end of the story. We embarked into systematically looking at this, and we culture cells from different background, Ivernaya, embryonic like fibroblasts, BJ cell, which is skin fibroblasts. We also used the adipocyte, and then we split them into contact inhibition only, serum withdrawal only, and we look at the transcriptomics. This is BAR. And also we found a data set that was using WI38. So this was not in our lab, in another lab that was published. And when we look at the gene set enrichment analysis, we saw that a lot of the inflammatory pathways were upregulated across pretty much all of these forms of quiescence, similarly to RS. So we also include sensitivity. So you could almost not distinguish between this was DJ cells RS, and these are IMR9 RS, these are pre-lymphocyte serum withdrawal. So they're different in terms of the individual genes that are activated, but in terms of property, they're very similar. We also did this in high oxygen versus low oxygen, and we noticed that certain forms of quiescence are more susceptible to high oxygen versus low oxygen. For example, serum withdrawal is less susceptible to becoming inflammatory if it is in high oxygens versus if it is in low oxygen. So oxygen also has a component here. This is all transcriptional. We started doing, we extracted the secreted protein, and we started doing panels of SAS. And as you can see here, contact inhibition shows a very large level of SAS activation, again, which is dependent on oxygen. Serum withdrawal is less so, but this indicates that these proteins are not just transcribed at high level. These genes are not just transcribed at high level, they are also secreted at high level in these cells that are in cell culture induced into quiescence. So what we did is, well, let's take this approach. This was a very small panel. So let's take this approach in an unbiased manner. And we look at the human protein atlas. And we try to, in an unbiased manner, identify all the genes that are considered as potentially secreted. So we found about 891 genes predicted to have at least one secreted protein. And we saw 723 upregulated genes in quiescence across all of our mutations. We call this the quiescence associated secretory phenotype. We don't know if they are all secreted. This is temporary. We know a few are secreted, but potentially about 723 proteins that are potentially secreted are being identified as upregulated in quiescence. And we compare that graph with what is being published at the SAP on senescence. We saw a tremendous overlap between the two. So a lot of what's potentially secreted in quiescence is very similar to what is secreted in senescence. But when we look at the specific in different forms of quiescence in different cell types, there's a lot of heterogeneity. So different combinations are expressed. I cannot say secreted because we don't have that. We're working with a mass spec collaborator to identify which ones are actually secreted. And the last thing I'll show you is this, which is when we looked at data sets that have been published in diseases that have a component to be associated with senescence, we wondered, is it all senescence? Or can we find evidence that also quiescent cells in those diseases show this high level of expression of potentially secreted inflammatory proteins? So we took an IPF data set from human, idiopathic pulmonary fibrosis. It was a single-cell transcriptomics. And we looked at the proportion of cells in different conditions, proliferating, quiescent, and senescence. Then we focused only on the quiescent one, based on our trained algorithms. And we split the quiescence into fibroblasts, and we also found a large percent of quiescent myofibroblasts that didn't show any proliferative marker. For each one of these cells, we defined a QASP score, which was the average of the expression of all the QASP genes in that cell. And what we saw that in healthy individuals, it's bimodal. We don't understand why this is bimodal. You see two different levels. The myofibroblasts tend to have overall a little bit higher level of the QASP. So myofibroblasts tend to be expressing these potentially secreted inflammatory factors more than the fibroblasts. But what is striking is what happens in IPF, in which you see a significant increase of the presence of myofibroblasts that has a very high level of quast, that is potentially secreted inflammatory proteins. So this is our first attempt to look at quiescent inflammatory state in tissues. And we're continuing with this, and we're trying to find this in other diseases that are associated with senescence, because we think that senescence is not the only inflammatory component. Quiescent cells might also be one of them. So to conclude this part, the last part is quiescent cell secretory phenotype. These quiescent cells exhibit a secretory program that is very similar to the senescence secretory program. It is relevant for disease potentially because are upregulated in non-senescent cells. For example, I showed you in fibroblasts and myofibroblasts in the context of IPF. I also showed you that this expression is very much dependent on the oxygen level. So this is another angle that we're looking at in terms of what is the role of oxygen in determining this response in terms of quiescent cells being inflammatory eventually. With this, I would like to conclude by thanking the people that did the work in my lab. Most of the work was actually done by people that are now alumni. Where is she? Azu Rocha did all the work about the quiescent cells, and Yvonne worked in the original paper from 2019. Anthony is a graduate student that developed the machine learning models, and Sam is a technician that is super skilled, and she's doing all the tissue analysis. And I would like also to thank my collaborators, Dr. Chandra for the initial studies, Stephen Wang, and John Ma for the study for the merfish, as well as the funding from the mostly NIH with the Senescence Consortium that was the nucleum. We have a big program that studies epigenomics, and we have time to talk about that, but we also look at these changes in the nucleus of cells. And this is our new center on the biology of aging. We are on the 6th floor, finally all together. For the longest time around, we were scattered across departments, but now the center has been consolidated in a single floor. We're all happy. There's a lot of interactions. Thank you.